Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Ted Zwang, Assistant Professor of Neurology at Massachusetts General Hospital and Harvard Medical School, and Jasen Zhang, a Biostatistics PhD student at UCLA. Today, we will be getting an update on their collaborations emerging novel neural recording devices with machine learning to decode Alzheimer's dynamics and cognitive decline. This effort was co-founded by the Kavli Foundation, Cure Alzheimer's Fund, and the Karen Toffler Charitable Trust. So welcome, Ted and Jasen.
Hi, Deb. It's great to be here.
Hey, Jasen. It's good to see you.
Hello, Deborah. Thanks for having me.
Wonderful. Well, I've been looking forward to this since we funded this last year. Can we just start with, tell us about your effort together and what was the hypotheses and what are you trying to prove? And really kind of explore what role each of you are playing on this collaboration.
Yeah, so this is a project that's been near and dear to my heart for several years now in different forums. And one of the questions we were really trying to get at is what is the trajectory of changes that we expect to see when pathology starts to affect individual neurons? Does it go from individual neuron to circuits to whole brain level to behavior? Or does it happen in some kind of other order? And is there something we can really use technology-wise to understand that and predict the kinds of changes we might expect to see in a person? As I've often heard from, my parents and my parents' friends lately, there's this huge concern that will come up whenever someone is diagnosed with mild cognitive impairment or some kind of early form of dementia. And that is, how many years do I have left of being me, of having my brain work? And, how can you tell someone what that is right now is a huge concern, both for doctors and for other, researchers that I know who really wish there was a better way that we could tell someone what we might think their trajectory is, rather than saying, okay, well, you're at the gate and we don't know what's through the gate. So what I started with were these kinds of electronics that we could implant into mouse models of the disease that are very flexible and mimic the structure and flexibility of neurons in the brain, with the idea that they could evade the immune response that typically causes these kinds of brain-machine interfaces to fail. In doing so, we were able to record the same activity of individual neurons in multiple brain regions over months. And that let us really look at how neurons in a relatively early young mouse that are somewhat healthy would change and become sick, and how the circuits would be affected and how their behavior would be affected. And one of the things that I realized in doing that is there were a lot of really simple analyses that I was able to do where I could see, oh, obviously this is going wrong. But if I wanted to really get at the nuance of it, if I really wanted to understand some of the crazy data that I was producing, and keep in mind, this is something like 15 million action potentials from thousands of neurons and multiple different animals doing different things, I needed to find some help. And this is where Jasen and Andrew come in. I got to talking with Andrew at one of your meetings a couple years ago, and it spawned this really wonderful collaboration where now we have Jasen starting to look at these data and develop statistical models to help us understand these data. And Jasen, you want to say a little bit about what it is you're doing with it?
Yeah. So, as Ted just said, this project was started about because my advisor, Andrew Holbrook, a professor at UCLA, Biostatistics, him and Ted came together and thought that this would be a rich area of biostatistical research because there are millions of action potentials collected over a long time period, which is ripe for machine learning techniques and statistical methodologies to decode these action potentials. After seeing millions of action potentials, what are these latent structures that are evolving over time? And that's the modeling that I want to do. Yeah, so it was amazing being able to play with the data set that Ted generously collected for me. And as a biostatistician, it has been a treat to analyze the data and build models to figure out what exactly this data is telling us. As a statistician, I like to describe my job as uncovering the truth of the story that the data is telling us. And the goal is to use the data set to see how Alzheimer's progresses over time through these mice models.
So, Jasen, how do you do that? Can you give us a visual on all this data? How do you even go about organizing it to be able to look at comparison or connections? How do you even do that? How do you even get started? Ted gives you all this data. How do you get started?
Yes. So the data set is massive. And the first thing I noticed is that there's different data modalities of neuroscience data. So for example, there's brain wave data, and that might be seen in like EEG or fMRI or local field potential data. But the subsection of the data that I was working with is called spike train data, and it just captures the action potentials. So the data set is just points in time in which an action potential fired for each neuron. So for example, a single neuron might have 300 firings throughout the 15-minute experimental session. So my data set would just be time points. So with that type of data, you can calculate firing rate. You can see if some neurons are silent, which means there are no firings whatsoever during that experiment. So the first things we looked at are how to summarize these behaviors. And neuron silencing and firing rates are pretty intuitive metrics of behaviors. That's how we started. And after we collected these statistics, there are correlative measures between neurons. So how do action potentials between neurons relate to each other? Does the action potential of one neuron follow immediately after that of another one statistically significantly? Or is there no correlative firing behavior? So that's what we're looking at.
So Ted, to you, when you got this data, when Jasen organized it and kind of showed you a little bit of what he was finding from this data, were there any surprises?
There certainly were surprises. I would say when you're analyzing data like this from the perspective of someone like me, I always think back to my advisor in grad school, Jackie Barton, who I remember would always be very skeptical of any kind of massive analyses or massive transformation or anything that needed you to apply an equation to see something that was real. So the first step really was, do we see anything that's obvious just from the data as it stands alone? And there were different ways that Jasen was able to take the data that I had collected and plot it so we could look at just some very simple metrics that would let us get a sense of how things were changing over time and assess whether, in our control samples, did it look like we were collecting information from the same neurons? Were there any differences that you could just see visually? between those two sets. And it was really crazy how different it looked when I saw that compared to how I had originally plotted it, where I did a lot of averages of everything together. So if I was to take the average of all of the neurons that were firing in the wild type mice versus the Alzheimer's model mice, it wasn't super interesting. It's the kind of thing where everything looked really stable. We might see some decreases in activity in the AD mice over time. But once we started picking apart what was happening to those individual neurons, that's when I would say the first real link clicked, that we had something that was incredibly cool. In our healthy wild type mice, the neurons were all very stable over the course of the several months that we were recording from. But in the mice with this tau pathology, we were able to see that several of the neurons looked like they were becoming quiet over time. And it wasn't enough of them that we could obviously see this huge difference in the average, but it was enough when you started to pick apart the individuals that we could see there were some really weird things that were happening that this tau pathology was causing. And I would say that that was one of the first big aha moments that Jasen helped to initiate, and that this collaboration really helped with. This idea that, we were able to just visualize this change and see a subset of neurons that looked weird. And it was only really a subset. A lot of the neurons in these mice looked perfectly fine, perfectly healthy. So that really kicked off a lot of what Jasen, Andrew, and I have been doing over the past several months to understand what is it that's different about these neurons that are becoming quiet. Why are they becoming quiet? What kind of brain activity is influenced by them becoming quiet? Is it just something where, it's happening to the individuals? Is it affecting the way they're talking to one another? And we've really taken this approach of starting with a lot of these great visualizations that Jasen did, seeing what looked weird, and then testing hypotheses about what we thought those weird things might be caused by and what they might be doing.
So you started with the discussion around seeing how the disease progresses and trying to predict, for somebody how much more time they have being themselves. Why did you pick the neurons to look at versus the cells inside the brain that might be fatiguing or dying? Can you go into that a little bit more between the relationship between cellular death and neuron activity?
Yeah, so the brain is composed of a lot of different types of cells. And I'm sure, over your course of talking with people, you've heard a variety of them that are implicated in Alzheimer's, that become weird, that might become dysfunctional. But one thing that you learn when you start to look at these different cells is they all have different roles. And some are easier to associate with memories. They're easier to associate with dementia than others. And a similar problem is some are easier to study than others. So I would love to have methodology that let me look at astrocytes and astrocyte activity over the same time course. I think that would be fascinating. And I would love to look at vasculature and see how it's changing over time. The problem is the kinds of methods to do that in the same way don't really exist. Even just trying to do this with neurons, I would say exists because there's something that's electrically active, which has allowed for certain kinds of methods to be developed over time, reaching to the point where I was able to make these flexible electronics that are specifically attuned to the activity of neurons and ignore everything else. So we can actually find how the individual neurons are changing over time. So in a way, it's a technological limitation that lets us look at these because of the importance for different learning and memory and so on that neurons have, these technologies have been developed much, much more than a lot of these other cell types. So, I think it would be really exciting to look at all of these other things. But to get to your other question about cell death, it's something that we would love to link to. How do these changes in neuronal activities lead to a neuron dying? But when we're looking at activity, it can be really hard to know that something has died versus thinking that your equipment has failed. So, at some point, you have to start with proving to people that you're able to do this to begin with, that in a perfect world, your equipment's not failing, that you are actually recording from the same neurons and from the same circuits, before you can make that leap to saying, all right, I'm going to try in a much more aggressive model of pathology where a lot of neurons are dying. Otherwise, someone's going to look at it and they'll say, oh, your recordings look like they're unstable. Maybe the equipment's failing over time, which is a very standard thing to happen when you're looking in the brain. So I would say that there's a lot of hope for future experiments to try and do these things, either with the technology we have to look at dying neurons or with new technology we're hoping to develop to look at things like vasculature and neurovascular coupling. But it's just not there yet.
So, Jasen, back to all of this data and trying to figure out the insights into this data. Were there specific kind of advanced technologies that you used? Did you use AI? What is kind of the state of the art that you're having to kind of explore to be able to use all this data to look for that change detection of what's going on inside the brain?
AI is definitely super trendy and super, like everyone's talking about it with AI, right? ChatGPT, all these LLMs. And AI has potential in so many domains, including medicine. But I think the biggest concern about AI and its use in areas like neuroscience is that the lack of understanding how these AI models get to their conclusions. So to say that I'm using AI would be misleading, but rather we're using classical machine learning techniques to re-represent the data in a more interpretable manner, right? We see millions of action potentials with their individual timestamps. We want to make sense of what is being described with these time points. So Andrew, my advisor, and I are developing graphical machine learning approaches to model the neural connectivity between all these neurons that collect the data for. And we want to see how the network of neurons changes over time. Like which neurons, which pairs of neurons are connected the strongest, and do they change over time? Do certain connections appear? Do certain connections disappear? And how do these changes in the network differ between the wild-type mice and the tau mice? Tau being genetically modified mice to be predisposed to Alzheimer's. So that's a graphical statistical approach that we're using, which I cannot say is called AI.
Does that exist already, or is this something new? Is this a new technology that you guys are developing to really kind of create this robust intelligence capability inside the brain?
From my end, the statistical modeling is just it's like an innovation from existing methods that we found in statistical journals. The small novelty that I guess there's two features to our model that is rather novel. The first feature is the longitudinal aspect. We are collecting data over several weeks, right? So we have the mice do the same experiment week after week for about 15 weeks. So this allows our data set to be longitudinal in nature, right? We want to see how neuron I and neuron J, how their connection changes over time. And that longitudinal modeling we haven't seen in past statistical literature. So that's our novelty there. Something else we are incorporating into our model is by conditioning on the mouse's behavior or the settings of the experiment into our analysis. In statistics, this is called like conditional models, where given that the mouse is running, how do the neurons fire? How do the neurons interact when the mouse is running versus when the mouse is resting? So you're conditioning on certain behaviors or certain settings, and then you build your model. So this conditioning, given that the mouse is running, how do the neurons look? Given that the mouse is in this specific maze in which they're running and they're getting rewards, given these parameters, how does the model look? So we're adjusting for these covariates, these confounders. And that's the second addition to our statistical model that we haven't seen been done before.
And I would say from my perspective, there's a lot of difficulty in analyzing data like this. So finding someone who has the skill set to be able to analyze these data and has, some kind of framework with which they can already start to imagine how to analyze this data is what makes this collaboration such a fruitful one. And while you might have a problem with the term AI, Jasen, I know from Andrew's perspective, he considers this an example of unsupervised learning and machine learning, which he finds to be a very vibrant subfield, with the goal of us trying to recover dependency structure from functional brain data. So I think, ignoring the nuance of what these terms mean, there's a lot of hope that these kinds of statistical methods that are able to take this massive amount of data and use it rather than how a lot of AI is usually imagined as like a black box, but use it in a way that has this kind of structure, really gives us a huge advantage for testing hypotheses for how brain activity influences other brain activity, both during the same time and in the future. And once we start to understand how these things change, how the trajectory changes over time for many of these different axes that the data is changing along, we can start to tease apart, okay, well, what is it that is the earliest sign that something's going to start to go wrong? What is it that fails after? And what are some signals that just happen to be coincidental, but don't necessarily correspond to our ability to predict future dysfunction? So right now, we're really starting with a very simple model as far as the biology is concerned, in part because these statistical methods to analyze it are already, at least to me, so complex that if we were to build in too much difficulty biologically, we might not be able to tease apart how all of these things influence one another. But that also means that as Jasen and Andrew build these models, we can also start to build out the biological model and establish how different co-pathologies, how other features may influence the trajectory that we are seeing. So one example would be including amyloid, or maybe including some other form of vascular dysfunction, maybe perturbing the system in a way that reflects menopause, or other features that are thought to be involved in someone's risk for having dementia, and see how that kind of perturbation is reflected in the models that Jasen is developing, and whether we're able to really tease apart how their trajectory is in a way we can relate to the human condition.
So if I understood that correctly, it's like it's a building block. You have this one block that you have and that you've looked at, but there might be some other building blocks that you go and you test that have, millions of points of data. But then you can combine those two things to look at. You wouldn't have to run the whole experiment all over with the added variables. You can do other tests with those variables and then add those building blocks together. Am I asking that correctly?
Right. I think so, but maybe a better metaphor would be thinking about, the development of television. So in this case, I'm creating a lot of data that I'm then streaming out to Jasen. And Jasen is trying to take that information and turn it into a moving picture on a screen that you can get something out of that visually shows you something meaningful rather than whatever, is moving through the internet or cable or, whatever you have. Now, let's say Jasen had never seen a TV before. And I told him, we're going to, try and recreate Game of Thrones. That would be a big ask. So instead, it's more like, all right, we'll start with black and white TV or maybe radio, something that's relatively simple. And then we'll start to build in all of these different features. And then eventually we'll get to the part that really tells the story that a lot of people are compelled by. And I think the challenge here is we really are operating from the standpoint of we don't know what the story is. We don't know where the story is going. We have some idea what the signals are, and we have to create some kind of, television to translate all of that information into the story. So it's tricky. And I think one of the ways we can approach it is by starting with what's obvious, things we can test that just kind of appear from looking at the data, and then start to look at the things that are less obvious that we really can only understand when we start to use these complex machine learning methods that are able to tease out what is going on in those signals that might be just incredibly difficult for an individual to look at.
Yeah, I like that analogy. It made it much more sense because it's just very hard to kind of think about all the different behaviors or just, conditions that you're trying to collect on. So I appreciate that. Ted, now that you've done this and this was something that you wanted to do, is this going to change kind of the direction or refine the direction of that work that you're doing in your own lab?
Oh, very much so. So I've been showing some of these data to other people in the field to try and get a sense of, what they think about this and what their understanding is. And I would say that a lot of them are very surprised by what we're seeing. One of the coolest things to come out of this, I would say, is the fact that we can look at these neurons and we can see ones that are on the cusp of dysfunction several months before they turn off. The other thing that's really exciting is we're able to see neurons after they've been affected by tau pathology, after they've turned off, after they've become dysfunctional, eventually reactivate. There are two reasons that this is very exciting. One is it goes against some of the conventional wisdom that once a neuron is sick, it's persistently sick, which gives us some hope that if we can figure out why the ones that recover do recover, we might be able to help promote all of the neurons to recover. And the other reason it's exciting is these processes, the fact that they happen over months, it really says that this is not something you would be able to see with conventional cross-sectional data. It's not something you would be able to see with a lot of standard methods for looking at the brain. And it's a real unique advantage to the technology that we've developed. So we can hone in on that and try to see, well, what kinds of new signals are we able to uncover that were previously invisible for how the brain is changing? And are there ones that we can see this far ahead of time that might be reflected in some of the information that we get from human brain studies? And if that's the case, we might really have a great way to interpret a lot of the kinds of experiments that people are doing in humans with much greater sensitivity. So right now, I kind of imagine it like, something's going wrong in a person's brain. You're recording everything in that brain and you're trying to uncover what kinds of things are different. But usually you don't have someone sitting in a lab for 6 months trying to see how their brain is changing. So we're trying to make a Rosetta Stone of sorts, where we might be able to find how many of these different dimensions that have change are reflected in those kinds of human measurements, and see whether we can create a profile that you could get from a single cross-sectional point in a person that lets us know, hey, there's something wrong that's going to progress going forward. And I feel like this is the kind of thing that's, really exciting to me, because it both establishes just a completely novel signal that we hadn't seen before and didn't know existed. The fact that it's recoverable means that if we start to find a way to look at this in people, we might have a new way to understand a person's trajectory, both what might happen prospectively, but also how a treatment might deviate from that prospective degeneration. And once you get to the point where you can look at a person and you can start to see how a drug or a therapy is affecting them positively or not affecting them at all, it can really promote a lot of more high-powered clinical trials. It can really promote this idea of personalized medicine. So I would say that, this has been really exciting for me, because it really says, hey, we might be at the start of something really cool that could allow us to do those things that could really help people.
Yeah. Why do you think they're recovering? Were they overworked and they got tired and they're being rested and they come back? Why are they recovering?
That's a tricky question. And I would say right now, my answer is very speculative, because we have only done experiments in these limited numbers of models. But my working hypothesis is that in these mice at young ages, we actually see what looks like their brain is going crazy in a way where there's what we would call hypercoherence. So you can imagine that you have a lot of neurons that are supposed to talk with one another to try and form memories and inform your interactions with the outside world and, keep a lot of your cognition functioning properly. Well, what happens if a lot of those neurons are firing in a way that you don't want them to? What does your brain do in order to stop that from happening? And what we're seeing is this turning off and turning on of neurons that corresponds in time with a change in the coherence of the brain, causing it to go from this hypercoherent state to something that's approaching more normal. So I almost imagine it like if you've ever put up Christmas lights, you know how you'll have just this whole array, or at least I don't know if new Christmas lights do this. When I was young, if you put up Christmas lights, there would be this time where sometimes it would stop working and you'd have to check each of the individual bulbs and see which one was having something go wrong. You take one out, you put one back in. You take one out, you put one back in. I think that what's happening here is something similar, where our brain is basically saying, hey, something's wrong. We're overactive. Can we take out a neuron, just tell it to be quiet for a little bit, see how it affects the brain, and then if everything seems fine, maybe put it back. And if something doesn't seem fine, maybe we'll just keep it quiet. I think that that's what we're seeing. And I think that this may be a really cool model for testing how tau pathology influences brain activity in general. So we're pretty excited to try and look more into this possible mechanism, but it's at really early stages.
No, I appreciate that. So, Jasen, you're a PhD student. Is this a part of your thesis? Where does this take you in your career of, your thesis, defending, and getting your PhD?
Yeah, I come from more of like a statistical modeling background. So, as a statistician, it's always humbling to be immersed in someone like another discipline's data set in this case, is neuroscientific data. So it was awesome to learn about what data types and what kinds of data we can collect, how these neurons are interacting with each other, and like hearing about this hypothesis that there's like a soft reset between neurons in case the brain is hyperactive, like what Ted just mentioned. So my contribution to this project has very much been like modeling. So Ted would explain to me what the phenomena that we want to like explain to me the phenomenon that we want to visualize and put in a figure. So how do we communicate these findings? And it's my job to like wrangle the data and to come up with these statistics that convey the message. So Andrew, my advisor, said that this project would be, this data set is huge, and there's so many directions we can go about modeling. Right? Modeling is a bit of an art and a science. You could introduce a lot of parameters, and the more parameters you have, the less interpretable it is, unfortunately, but it's more flexible, right? It's more flexibility. You have less idea of like what each moving part is about. Whereas if we use a graphical approach, then a graph is connecting nodes with edges, right? If each neuron is a node, and we connect an edge to denote that there's some communication between these neurons, then that's a lot more interpretable. So this data set can give rise to several different modeling approaches, and that's what a PhD will be about, right? So this is my first idea, to like model the neural dynamics in a graphical manner over time longitudinally instead of just cross-sectionally, which was what was done before. So I'm very excited about this data set, and there's a lot of other directions that I've been thinking about how to take this, but for the next few months, I'll be proving these statistical methods and applying them to this data set. So yeah, slowly working on my PhD, which is very much about this data and this problem.
Yeah, no, I would imagine that it's, oh yeah, well, it's fun, right? You guys have collaborated in a way that, very few actually get to collaborate. So I think that's super exciting, and it's really great for both of your careers. Ted, if you kind of speculate, it sounds like there could be a diagnostic that comes out of this. It could be maybe even a somewhere along the way a therapeutic that comes out of this. I don't want to put you on the spot, but where do you think this goes in this work that you're doing in the next 3 to 5 years, and what help do you need? There's people out there wanting to help you too.
Well, so what I would say is, right now, there's a lot of mystery about the trajectory of individuals. And more than anything, I really think that, what I'm hoping to do is find some kind of biological signature, in this case, electrical activity in the brain, that lets us look at the status of a person and see what their trajectory might be. And I think that, as you said, the implications of that are really phenomenal. The challenge is always, how do you take these kinds of experiments that we're doing in mice and establish that these are happening in people? And how do we draw that kind of translational science from these preclinical models to the complexity that all of these individuals have in their brain? So I think that the first step for us, we're really trying to look at what's happening in ways that you can't look at in a person. But with the view that there's a lot of really incredible technology that's being developed right now to look at people that doesn't necessarily have a signal in mind that they're looking for. So what we're really hoping for is, if we can establish signals that we believe could translate to people, can we then find technologies that are best suited to those kinds of signals and collaborate with people, other scientists, other doctors, that would be able to try and look either in the data sets that they have or with new prospective technologies that could be developed to try and target those types of signals. So I think, it's not a short road. We're trying to work as quickly as we can. But I think that what's really got me excited is, I talked with earlier today, like Heidi Jacobs, who's doing this really wonderful MRI work in people. And, I look at all of the stuff that's going on there, and they're still doing some really incredible science to try and understand early diagnostics. And I'm really hoping that at some point we're there too. So I think that for me, at this early stage, one of the questions that we have is going to be, what kinds of signals can we translate to people? The other question is going to be, what are these co-pathologies doing? And then we're starting to talk with some other scientists. We recently talked with Tom Montine over at Stanford. This was something set up by Brad, where we want to see, all right, well, we have this kind of trajectory going along where we can see these changes. Can we take, a drug that another lab is developing and actually observe improvements at the level of these individual neurons and their circuits when we provide some kind of treatment? And is it something that we can monitor the trajectory of and predict the trajectory of? And I think once we establish that kind of causality between a drug and these signals that we're looking at, we'll have some really huge potential for doing the same in people. What would be a success in a person is challenging. But I remember hearing someone else say, the elephant in the room when it comes to studying people with dementia, when it comes to, people that come into the clinic, is at the point that they're there, their brain has already failed to the degree that they're starting to see effects on their behavior. And it might be really hard to bring that back. But if instead we have a better way to look at people earlier and establish some kind of way to look at what's going wrong and quantify that, we might be able to really treat them at a stage where they're easiest to be treated. And that's not to say, we don't want to work and help people that have already progressed substantially. But I think the hope is, even for those people, if the readout that we have is something where the neuron activity changes and tells us, hey, your brain is functioning much better, from 1 week to the next, it'll make them much happier than waiting for, a couple of years to see how they're doing on a cognitive array.
Yeah. Well, this has been just really fascinating and wonderful. What are the next steps? You've got this project, and I'm sure you're going to need to publish it and, get it out there and get it reviewed and such. But do you have those next 2 or 3 steps that you really, really want to do and try to get funded so that you can continue on this work and continue to work with each other?
Yeah. So I would say, from my perspective, one of the 2 next steps really are looking at co-pathologies and seeing how they deviate this trajectory of neuronal activity, with the hypothesis that there are some things that initiate the silencing of neurons, and there are other things that might prevent the recovery of neurons that become silent. And I think it'll be really exciting to see if, for example, amyloid, which seems to cause hyperactivity, results in an increased rate at which the neurons become silent. Or if there are other things like glial interactions that perturb the ability for these neurons to recover. And that could really help us flesh out how a lot of these things play with one another. And the other side is really going to be, can we look at these kinds of treatments and see how our metrics change as we treat a diseased brain? And at least as of right now, neither of those projects are funded. So, it's certainly the kind of thing that we would be looking towards in the future. And fortunately, regardless of how we do these perturbations, assuming we do them in very, similar ways to the original recordings we gave Jasen and Andrew, they can start to add these into their models and, develop some really clever insights into how the brain is networked and how it's changing.
Yeah, that's fascinating. Jasen, do you have any following thoughts there as we kind of wrap up?
My thoughts actually were something I learned throughout my time on the project, which is that the future steps are like a big chunk of like the future directions are dependent on what you see in the results that you just got. So what Andrew likes to say is that make these models and then we can figure out how to improve our modeling given what we see. So it feels like a large piece of investigative work. Like, what are the leads that our initial modeling gave us, and how should we best move forward seeing this? So this uncertainty used to not sit well with me, but as I've been on this project, it's definitely a hallmark of what a PhD is all about. This big question about neurodegeneration is not easy whatsoever, and it takes an adaptive mindset and clever modeling strategies and interdisciplinary work to get this all going. So for now, I will stick with, I'm in the middle of developing this graphical approach, but there's so many other ways than just this. But once we get this model working and we see how these graphs evolve over time, then we can plan our next steps. But yeah, a very much adaptive timeline.
I love that. So Ted, you get the last word here.
Well, I think the last word that I would want to say is just to kind of reiterate one of the reasons that I find this so exciting, which is really this idea that we have opened a new window into the brain to see a feature of neurodegeneration that we didn't know existed before. And it's one that gives me a lot of hope because it's something that says, hey, it's not just that things are shutting off, but some of them are turning back on. And I think that there's something to be really excited for when you see that you have some kind of process in the brain that is allowing for the recovery from something that has been causing problems. So I'm really just so excited to have seen that and to understand what it is that's happening and how we can take advantage of that is going to be, I think, a really promising avenue forward for helping people's brains get back to normal. The goal that we all have is, how can we help people? And I think that we're on a very fruitful thread that I think will really help people. So yeah, I'm so excited. This has been such a wonderful year in terms of this project, just seeing how, all of that data that we collected, that we've spent so much time collecting, actually gives us this really exciting new signature to look for.
Well, I'm excited for you both and what you're doing and the new findings and what's happening in your careers. So I'm very, very excited for that. And I really appreciate you giving us the update of what you're doing. And we'll look forward to getting more as time passes here. But congratulations on this. And we'll check in another 6 months, 8 months and see where you're at.
Sounds great. Thank you, Deb.
Thank you for having me.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.